I want to thank the organizers for giving me the possibility of presenting my work. So the title of my talk is Localized Enzymes Reflect Cellular Metabolic State. And so we've been talking a lot about networks, and also Triadic was saying, you know, what do networks really look like? And I guess sometimes we forget that the most known network is the metabolic network. Even before the genome, we already had this network. And somehow I feel that sometimes we forget about this network because we think it's already solved. We know all the connections and we know how all the enzymes you know, process the different metabolites. But so I wanted to ask a different, a bit go further with the idea of this network. So the question is, you know, you have all these reactions happening inside a cell. So how is this really organized? What does when you open the lid of the cell, how do all these enzymes look? And you know, the usual idea of how a metabolic reaction works, so here if you have, you know, 3 sets in a reaction, is that all the enzymes are basically homogeneously distributed inside the cell. But there could be a possibility where actually there's, you know, some structure in these reactions, and that depending on if the reactions, the enzymes are active or not, there could be changes in the structure. So this was a hypothesis that I kind of was eager to prove. And I was lucky enough to find that, you know, there's all the enzymes of E. coli are tagged with GFPs. So you can just go and look at what they look like. And as you can see, in a large subset of these enzymes tagged with GFPs, you can see that 25% of them show some, what I call localization, meaning that instead of being just homogeneously distributed, you see them localized at different places inside the cell. And this, you know, it's very varied, different enzymes show this pattern inside E. coli. And so actually, if you look at, you know, different pathways and try to see where these enzymes are, so for example, this is a PPP pathway, and, you know, if there are other examples. And you can see here in green basically which of the enzymes are localized in these pathways. And here you have the images. So you see that this doesn't really seem to be any rule of which of the enzymes are localized. And one of the things you have to think about it, these are E. coli cells grown exponentially. So the idea is that, you know, there's some commonality in between the metabolic processes going on in these cells growing exponentially. But you also have to think a bit about how pathways are defined, and, you know, actually, as we were discussing, you know, just before, you know, should we still talk about pathways? And in a sense, you know, a pathway is just defined by basically either the product it's doing or basically the genetic identity of a pathway. But you can see pathways in kind of a different way also. And but before that, one of the things we wanted to see is, you know, what was the proportion of, for example, pathway length to the number of localized enzymes that you find? And actually, what you see is that there's a nice scaling between pathway length and the number of enzymes. And actually, what you find is that, you know, for every basically 7 reactions, there seems to be one enzyme that localizes, that shows this, you know, very punctuate, focalized distribution. And so, as I was saying, you know, pathways are defined in a, let's say, biological way. It's not always very clear how pathways are defined. And in order to try to find, you know, some order or some similarities into which enzymes are distributed, I decided to divide the pathways into something that makes a little bit more sense, which are these elementary flux patterns. So if this is a pathway, you can actually subdivide into linear pathways, and that, you know, are the intersection between the definition of a pathway and then actually fluxes that are stable through these linear pathways. So once you do that, you can now basically identify an order of the localized enzymes. So for example, this one is localized in the first reaction, this one is localized in the second reaction, this one again the first reaction, this one again the first reaction. So when you do that, and you decompose all the reactions of E. coli, of all the pathways of E. coli, and you basically what you do here is you just look at, divide them by the number of steps there is in an EFP, one of these pathways. So it goes from, you know, 3 to 12. And you look at what's the proportion of enzymes that localize either in the first or in the last position of these reactions. What you see is that there's up to 60% of all the reactions show a localization of an enzyme either in the first or in the last step. And you see that there's a huge enhancement here in the reactions that have between 6 and 10 steps. which is statistically significant compared to just a null hypothesis here in red. Also, if you look at it's the same reactions that show localization in the first and in the last reaction, you see that there's also, of course, it's less because it's less probable that two enzymes are going to be localized in the same pathway, but up to 10% of the reactions show localization in the first and the last step of the reaction, and you see that there's also an enrichment for the reactions that happen between 5 and 10 steps. So basically, what I just showed you is that there seems to be a high preponderance of localized enzymes in E. coli cells grow exponentially, but also there seems to be a rule where the first and the last enzyme of these pathways localizes. So now the question is, where do those enzymes localize in the global metabolic network? And so this is the adjacency matrix, basically, of all of these reactions. So here you have reaction versus reactions. And so each dot means that one of the reactions is connected to the other. And in green, you have all the localized reactions. In blue, you have the non-localized reactions. And in red, you have connections between localized and non-localized reactions. So when you look at that, you see that by looking again at the degree of each of these nodes, you see that there's really no difference between the localized and non-localized reactions. But when you start looking at things that are a little bit more related to flux, so for example, with that matrix, you can calculate just the 3 loops or the 4 loops by just measuring the diagonal and multiplying the matrix, elevating the matrix to the cube or to the 4th degree. And you see that here you have a distribution of the number of counts of 3 loops for localized enzymes in green and for non-localized enzymes in blue. And so what you see is that for these cases, all the localized enzymes seem to have more 3 loops, 4 loops, and feed-forward 3 loops and feed-forward 4 loops. So in a sense, there's a higher density of flux around those enzymes, those localized enzymes. You can actually do some statistics on that. So if you compare the number of loops of localized nodes versus non-localized nodes, you see that there are statistics that are significantly different from non-localized enzymes by a KS test for 3, 4, 6, and feed-forward loops. The other thing you can do also is you can compare the distribution of all the localized enzymes to a randomized set of the same number of localized reactions, but now chosen randomly. And and you can repeat that and get a distribution and calculate a p-value to see how significant this localization of enzymes is when you compare the different loops. And so what you see is that, again, 3 loops, 4 loops, 5 loops, and the feed-forward loops are statistically significantly enriched when compared to just when you choose random nodes of these enzymes. One other thing that you can calculate is how connected the localized enzymes are between each other, and also how clustered they are. And so when you compare to how connected they are within each other, you see that they're more connected than the non-localized enzymes, but paradoxically their clustering coefficient is lower than the clustering coefficient of the non-localized enzymes. So what does this look like? So basically the clustering coefficient is higher, so it means they have more triangles, they're more connected to each other than the localized enzymes, but the within group connectivity is higher for the localized enzymes, so meaning that they're kind of forming something like a tree, so something that has a lot of connections, but the connections are not clustered. This seems to be a bit paradoxical with what I was telling you before, that there's actually more loops encompassing localized enzymes, but actually what happens is that there's more connections between localized enzymes to non-localized enzymes, and hence accounts for this higher number of loops for the localized enzymes. So if you think about it in the context of what I was telling you before, that in a sense, because when you take pathways, the first or the last enzyme of the pathway are localized, in a sense, the localized enzymes are defining where a pathway starts and where a pathway ends. So if you go from a node of an unlocalized enzyme and go through the node of a localized enzyme, what actually you're doing is you're defining a pathway in a sense. And so knowing that the localized enzymes are more connected, you're going to be closer to another pathway defined by another localized enzyme. So in a sense, the localized enzymes are doing are making smaller the whole network, the whole metabolic network, by just being more interconnected and having more loops with the non-localized enzymes. So I just gave you all this kind of structural properties which are kind of telling you that something is going on with these enzymes being localized in the cell. But why and when do these enzymes localize? And so when you again look at the gene ontology categories, you see that for the localized enzymes, essential processes that you'd expect to be enriched when cells are growing exponentially are very present. So things like nucleotide binding, ATP binding, amino acid binding, and ubiquitin synthesis, biosynthesis, and ion binding. So these processes are essential. So what you have to think is that basically these cells are growing exponentially and have this set of enzymes that are localized, and basically are localized because they're needed to be active for growth in these cells. So in a sense, they mirror the biological activities that are needed for growth in exponential cells. So what I did next is I took an example to see how this was working. So I took the cell wall synthesis pathway, which is a well-known and defined pathway in bacteria. So basically it's a linear pathway, and I tagged all the enzymes in this pathway. There are several steps, there are up to 10 enzymes. But what you see is that when you tag the first enzyme with a blue protein and the last enzyme with a green protein, what you see is that in cells growing exponentially, you see that these enzymes again localize, but also co-localize. So they seem to be kind of forming a complex together. And if you look at these same enzymes, but over time, where you're going from exponential growth to stationary growth, so stationary growth basically means that the cells are not, basically not dividing anymore. So you see that the numbers of cells that are showing localization of the enzymes go from being very present to almost being, absent. So you kind of see these, that as cells go into stationary phase, the number of cells that show localized enzymes goes down. So in a sense, this process of localization is also dynamic and is reflecting the biochemical processes that are going on in the cell. When the cell needs to fabricate a lot of cell wall, these two, the first and the last enzyme of this pathway, are localizing. But when the cell stops growing and stops fabricating cell wall, the enzymes are diffuse again and don't show this localization. So in summary, the idea is that if you have a pathway like the one I just showed you, and if you're in stationary growth, you just have homogeneously distributed enzymes, like we all think about these enzymes in a cell. But when you're going exponentially, there's kind of like a localization happening of the first and the last enzyme, and probably this influences all the rest of the reactions in the sense that they are localizing a specific position in the cell and probably make the reaction more effective. Also, these are some short-term images just showing that the whole complex is pretty dynamic, and you can see it moving around as probably synthesizing cell wall in the cell. So again, just take home message. There are a subset of enzymes seem to be localizing. They have very specific properties, where in general the first and the last enzyme of pathways is being localized, and they are interconnecting the localized enzymes to the non-localized enzymes. And so I think this property of bacteria probably holds also for eukaryotic cells. So I think there's a big advantage to try to take here and try to go further into the understanding of networks by trying to measure in single cells the activity of the different members of a network. And so I think in metabolism it has been a big problem to try to measure in single cells what's going on. But I think that imaging techniques can really bring a lot more dynamics and more information about how this is happening. Thank you. I just want to thank Gustavo Tolowitzki, which got interested in this work and which I have a lot of passion with, and Guillermo Cecchi also from IBM, who was also very eager into following these ideas and some of the experiments that I did at Columbia University. So thank you.
Yeah, really, really fascinating. Thank you. I wonder, have you thought about, instead of looking at first and last steps from this sort of graph theory perspective, have you looked at thermodynamically committed steps in these pathways and whether that bears any relationship to localization?
Yeah, I mean, initially what I had looked at was really kind of like what kind of enzymes are being localized. Are these enzymes that have, you know, lower KCATs or things like that? But I didn't see anything that was really showing a specific property of that. Maybe that can be revisited, but really, that's the, yeah.
Interesting. Thank you.
Yeah.
So this question is sort of related to the previous question. Usually the first enzyme in the pathway is thermodynamically important. As far as I know, the last is usually not. So I was wondering, is it the case in the elementary flux patterns that the last enzyme in the pathway is also the first enzyme in some other pathway?
I guess it happens, but it's nothing preponderant either. I mean,
Okay.
nothing that kind of stood out. Yeah.
And one other question was, I didn't see glycolysis as being enriched in localized enzymes, but if you have exponential growth, it should be upregulated, but maybe it's just so prevalent that it can't be localized? Is that?
No, yeah, glycolysis was there. I
Oh,
think.
it was there?
Yeah,
Sorry.
it was one of the first pathways.
Okay.
So you see, yeah. You have a bunch of enzymes being localized there.
Thank you for the talk. I have two questions. The first one is, how penetrant is the expression of the enzyme? Because in yeast, sometimes when they are in a compromised condition, you remove one of the amino acid, it won't be 100% of the cell that will express the enzyme. There will be 10 to 40%. So the same thing in bacteria. And also the second question is, those enzymes that are fork point, for example, RO2 in yeast, which is the enzyme that produces charismate, is the fork point of all the aromatic amino acid production, and this one is localized in yeast. Is it the same thing in E. coli?
So I also looked at initially branch points, because as you see that here, it seems that some of the branch points seem to be localized. But again, it really seems to be that it's only in the context of flux that these localizations really have a meaning. So I didn't see any enrichment on fork points. In terms of the first question, again, just experimental, you have to think of this as, so all these enzymes are basically just very lowly expressed because they're under an IPTG promoter, but there's no IPTG, so it's really just kind of like read through those promoters. So that's in a sense what's interesting, because if you're overexpressing these things, you can get a lot of artifacts and things like that. So that's really you don't want to deal with. And I think there's so from the images I took with these cells, there's a lot of variability. So as I was showing, maybe 60% of the cells show this localization at the maximum. So again, I mean, probably there's a lot of variability in terms of metabolism of the cells. And I think those are also interesting questions, right? We are all interested in kind of how noise regulates signal transduction pathways, but of course in metabolism it's probably going to be something similar. So small differences in quantities of metabolites can change the metabolic rate of the enzymes and of the cells. So I'm sure there's also metabolic individuality with cells. Thank you.